today we are going to study pathogenicity and virulence. I am Dr. Sharad Deshmukh from S.H. Jaiswal College, Arjuni Morgaon. So, we will start the pathogenicity and virulence. So, first let us see these two terms, what is pathogenicity and virulence. Pathogenicity means it refers the ability of a microbial species to produce the disease. Here one thing you remember, the term pathogenicity is related to the species, while the term virulence is related to the strain of a species. So, virulence means or it refers the ability of a strain of microbial species to cause the disease. So, strain of a microbial species is related to the virulence. The best example we have mentioned here, the example of the M. tuberculosis and poliovirus. In M. tuberculosis there are pathogenic one, but there are some strains which are less virulent, which are more virulent, or some are the avirulent. One of the avirulent strain of the M. tuberculosis is the B.Sc strain, which is used for the vaccination. In this way, in number of the microorganisms or in the bacteria, there are the certain strains which vary in their virulence power within a species. So, that is why you should remember what is pathogenicity and virulence. Now, let us see the two terms attenuation and exaltation, because these are related to the particular strain or a species, and the term attenuation means to reduce the virulence power of a strain, and that is known as the attenuation. So, attenuation you can simply remember to reduce the virulence of a particular strain. Then how to reduce this virulence? So, it can be done by various methods. One of the method is repeated passage through the unfavorable host. That means, if the particular strain of a microorganism is inoculated in unfavorable host, which is the not susceptible to that microorganism, and it is passes through this host, that may become avirulent. That is one of the way to obtain the avirulent strain. Secondly, repeated subculture in artificial media. This is the most simple method, is used particularly in the BCG vaccine preparation, where the strains are repeatedly cultured on artificial media for number of times, near more than 200 times they are subculture, and during this period the strain may lose their virulence, and that you will get the avirulent strain, and that can be used for the vaccine. Similarly, the third method is the prolonged storage in the culture. Every time we used to prepare the cultures and we are storage in the low temperature, but if you store it for the longer period than the period generally we use for subculturing, that can result into the loss of virulence in the microorganism. So, prolonged storage is one of the way to obtain the avirulent strain. The next is growth under the high temperature. High temperature means it is not so much high, but it is the temperature which is higher than the optimum temperature required for the growth of the bacteria. So, generally we are growing the bacteria at 37 degrees centigrade for most of the bacteria. So, if slightly higher temperature you use, about 40 to 45 degrees centigrade, at that temperature they may lose the virulence by growing them at the higher one or higher temperature. The next one is the growth in the presence of weak antiseptic. So, if you add some antiseptic solutions in their media, and if you grow them in presence of this antiseptic, that may also result into the loss of virulence of the microorganism. So, these are the various ways by which you can obtain the attenuated strains. Attenuated means those which have lost their virulence power, but they have retained their antigenicity. That means they are able to induce the antibody production or induce the immunity, but they are not able to cause any harm to the human being. That process is referred as the attenuation. Now, the next process is the exaltation. So, it is just opposite to the attenuation. So, it is an enhancement of the virulence power of a strain is called the exaltation. So, you must remember these two terms, attenuation to reduce the virulence, exaltation to enhance the virulence power of a strain. So, in this way these two terms are commonly used. Next, we come to the infecting dose. So, an adequate number of bacteria should gain the entry in a host for successful infection, and which is estimated as MID, that is minimum infecting dose, and MLD, that is minimum lethal dose. So, when the organism enter into the host, so they should get the entry in the minimum number or the at least that number which is able to establish the infection, and once infection is established, that can result into the disease. So, then and only then they can cause the harm to the host. If they are not in the very low number, that may not able to cause the successful infection and ultimately may fail to cause the disease. That is why they have estimated the term MID and MLD. Let us see these two terms. First, we will see about the minimum infecting dose, that is MID. So, it is the minimum number of bacteria required to produce the clinical evidence of a disease, that is known as minimum infecting dose, while minimum lethal dose, that is MLD, is the minimum number of bacteria required to produce the death in a susceptible animal under standard condition. So, these two terms you must understand clearly. MID means only the infecting dose able to produce the clinical evidence of the disease, while the lethal means to produce the death in the experimental animal, susceptible experimental animal under the standard condition. These doses are estimated as ID50 and LD50 as the dose required to infect or kill 50% of animals tested under the standard condition. So, instead of only MID and MLD, it is estimated as ID50 and LD50. ID means infecting dose. So, you can clearly understood from this as the dose required to infect the 50% of the tested animal under standard condition, that is the ID50, and LD50 means as the dose required to kill the 50% of the animals tested under the standard condition. That are these two terms are used, ID50 and LD50. Next, we will come to the various determinants of the virulence, or sometimes it is referred in number of books as a virulence factor. Both are the same terms, virulence factors or determinants of the virulence. That means the factor which are determining the virulence of a causative agent or infecting microorganism. So, this includes the adhesion, then invasiveness, toxigenicity, enzymes, anti-phagocytic factor, survival within phagocyte, infecting dose, genetic factor, route of infection, communicability, that determines the virulence of a particular infecting agent. And if they have some determinants, they may possess one or more than one among these, then they can establish the successful infection in the host and can result into the disease. So, we will see one by one. The first I have mentioned already, the adhesion. What is the adhesion? It is the attachment of the bacteria to the body surface. The term itself suggests adhesive property. That means it can able to adhere to the surface in the body, that is known as the adhesion. And how they can adhere? So, it is the specific reaction between the surface receptor on the host cell. So, host cell having the surface receptor and adhesive structure which are present on the bacterial surface called as the adhesion. So, this is the interactive mechanism where the host cell surface get adhered to the adhesive structure which is present on the causative agent, that is the bacterial cell, and that is referred as the adhesion. So, you must know the term what is adhesion and the adhesion. The phenomena is the adhesion, where the adhesion is able to act or react with a specific receptor on the host cell. Adhesion generally occur on the fimri or the pili. This is the most important structure present in the bacteria, that is the fimri or the pili, which act as a adhesive factor or as a colonization factor. In addition to this, there are some other structure which plays the important role in adhesion. These include the flagella, outer membrane protein, and lipopolysaccharide, that is LPS. These also act as a adhesive factor. If they are present in the bacterial strain, they are able to adhere very smoothly to the host cell and they can establish the infection easily. So, the adhesions are generally made up of the protein, that means they are protein in nature, and as they are protein in nature, they are antigenic in nature. Generally, the antigen is the protein in nature, and that is why these adhesions factor which is protein in nature, and that is why they are antigenic. Antigenic means they are able to induce the immune response in the host. So, the specific immunization with adhesion can be attempted as a method of prophylaxis in some infections like E. coli causing diarrhea in cows and gonorrhea in men. So, in these diseases, the specific antigen can be used for the prophylactic measure against this infection, because if these agents are inoculated, that will induce the immune response and ultimately they will prevent the infection from the E. coli or the gonorrhea in the man. So, we will see the next virulence factor, that is invasiveness. This invasiveness means it is the ability of a pathogen to spread in the host tissue after establishment of the infection. So, when the pathogen establishes the infection, it should be able to spread in the host, then it can cause the severe infection. Generally, those pathogens which are highly invasive are able to cause the serious infection as compared to those pathogens which are less invasive. So, the invasiveness power, if it is much more, and that will cause much more severe infection. So, you can see here the highly invasive pathogens produce the spreading or generalized lesion. Example is the streptococcal septicemia following the wound infection. Here the streptococci are able to cause wound infection at the specific site, but they are spreading or causing the generalized infection, and they are spreading in the blood, and they are multiplying, growing, and produce the toxic product. So, that is why it is known as streptococcal septicemia, although they are entering only through the wound. So, this is the invasive power is much more in the streptococci than the staphylococci, that is why they are able to cause the septicemia. While in the next case, you can see here the less invasive pathogens cause the localized lesion. The example here I have mentioned is the Staphylococcus aureus. When it infects the wound or the cuts, then it causes localized lesion, or in the skin they cause the very small lesions, and they are not spreading much more throughout the body at the specific site on the skin, like boils, abscesses, or the wound infection caused by the Staphylococcus aureus. Here we have already mentioned, if they are more invasive means more serious infection, if they are less invasive means very less severe infection. But there are certain exceptions to this. Although there are some pathogens which are not having much more invasive power but they can cause very serious infection. Now you can see here some pathogens lack the invasiveness but can cause the serious infection. The best example is the Clostridium tetani, which remains confined to the site of the entry, but the tetanus caused by the Clostridium tetani is very serious disease. If it is not treated, that result ultimately the death of a person. So although tetani remain confined to the site of the entry, that is wound infection, but they are able to produce the powerful toxin which spread, which act on the central nervous system and cause the harmful effect to the host. And that is why although they are not invasive, but they are highly pathogenic and highly active. So this is one of the exception for the invasiveness. Now we will see the remaining part in the next lecture. Thanks for watching this video, and next video will be uploaded soon.